bit now. I'm Julie Goodley, I'm Director of the Lister Institute, and it's a great pleasure that we've got a panel up here for you to do some more question and answering. You've already done that really well already, but we've got two more fellows on the panel for you. We've got Chris and Lizzy. So first of all, can I just get you to introduce yourselves?
So I'm Chris Hill. I won the Lister Prize last year. I'm based at the University of York in the UK, and I'm a structural biologist interested in molecular mechanisms of RNA-protein interaction and viral infection.
Hello. Oh, very loud. Sorry. I'm Lizzy Roster. I'm at University College London. I'm a B-cell immunologist, and I'm interested about how B cells contribute to inflammation in autoimmune conditions, but particularly childhood onset autoimmune conditions.
So whilst you're thinking of questions that you might want to ask the panel, I'm going to start off by asking Lizzy, what strategy did you use when you were kind of coming up with your Lister proposal?
I have a senior research fellowship from the Kennedy Trust for Rheumatology Research, and that project's about B cells and inflammation in juvenile idiopathic arthritis, and really about the gut-joint axis, so how changes at the gut, so the microbiota can cause impact B cell development in that condition. When I was developing that application, it became aware through discussions that lots of children with arthritis also develop uveitis, which is an inflammatory disorder of the eye, and that's what my project, my Lister project, focused on. As part of that process, people kept on saying to me, the eyes seem like a really big opportunity, and because nobody was really kind of doing basic immunology in that place. It was incredibly high risk as well. It's very difficult to access eye tissue from children with arthritis for obvious reasons. So it seemed kind of like the perfect opportunity to give it a go because it was high risk, and I've become increasingly passionate about working with that patient population. It was also on the back of an unsuccessful Lister application in 2019, talking about failures, where I think I was developing a project that wasn't high risk. I hadn't really built up my profile in the right way, and it wasn't really, I think, at the cutting edge of research. So I used the opportunity at that time. I thought, well, I just got my Kennedy, it seemed like a big deal. And everybody kept on saying somebody needs to answer this really important clinical question in our patient cohort. So I gave it a go, and here I am now.
Thank you. Chris, picking up on what Aga was talking about earlier, do you want to just describe how you were mentored when you were putting your application in last year?
Yeah, absolutely. So the first thing, I think, the best advice I was given, which was from my deputy head of department, who was the head of research in the department, was this is a charity that funds important medical unsolved problems that matter. So you have to be able to tell a room of non-experts why they should care about your question in 5 seconds, basically, or 10 seconds. Why is this an important question? Why is it worth investing in? And that's perhaps a slightly different framing for a research application than you would, say, approach MRC or Wellcome Trust with, where it's a long form program and there's a lot of basic discovery research that you can go into detail. But for me, that was really useful mentoring. Then during the application process itself, I was very fortunate in that I reached out to a lot of former Lister fellows and current Lister fellows to ask, well, would you be willing to share a copy of your application? Would you be willing to share your slide deck? Would you be willing to, what's the interview process like? What do you think of this idea? And more than half of them replied. They were really, really helpful in helping me refine my ideas. And then the third component of it really, I think, was good preparation. So I had at least 4 or 5 different people read my application and suggest, quite brutally actually, whole paragraphs were crossed out, or I'll remember one comment in Track Changes from a senior cancer scientist who was not in the field. She just highlighted the whole opening paragraph and put bland. But it's that super honest feedback that you need, because if someone on the panel thinks it's bland, it's not going to get shortlist. And then the mock interviews, and my goodness, they were terrible. There were some of get the scariest people at your institution in the room, round the table, people who chair MRC panels, people who sit on scary decision boards, and get them to put you through the ringer on your research. Not only the methods, but the conception of it, why it's an important medical problem. So, for me, I was proposing to work on enteroviruses and how enteroviruses hijack ribosomes. And question 1 in my first mock was, well, there's a polio vaccine, so who cares? Bye. Like, no one's really affected by these diseases, are they? So immediately you're on the back foot and you're having to justify, hone, defend your ideas and your approach. Another key point outside of a formal mentoring framework where people are looking at your application, people are giving you mock interviews, I think is engaging with experts in the technology space that you operate in. So for me as a structural biologist, and this goes beyond the tenure of the Lister application, but it goes throughout my whole career really, I finished my PhD in 2015 and I trained in X-ray crystallography. But in 2015 was when the direct electron detector was invented and when the Titan Cryos microscope hit the mainstream for Cryo-EM, and we started seeing atomic resolution structures coming out of Cryo-EM. And this was massively disruptive, right? You could work on difficult multi-protein complexes that you couldn't purify much of, you didn't need to grow crystals. And so I decided to retrain then, and I talked in detail with the technical practitioners who are expert in this when I was thinking about what research questions can one actually answer. And then again, the big pivot for me for my Lister application was moving to the forefront of what it is possible to answer. So it's now not just using Cryo-EM on in vitro samples, but actually using it in situ to look at structures within infected cells at atomic resolution, which is now technically feasible. Now, I didn't have much preliminary data, but as part of the application, I was talking constantly to expert practitioners in this technique, so people in the Leeds facility who I'd proposed to work with, and they were saying, well, you can't use this cell type because they won't spread out thinly enough over the grids, but you're going to have to do this, you won't get good enough resolution here, but you will there. And so it was sort of a co-development of the whole application, but informed by technical best practice. And that's important because I didn't have any preliminary data, so I needed to know what I was proposing was feasible. So just a few ideas there.
Thank you. So have we got any questions from the floor?
Speaking from the Irish perspective, and I have to say, I didn't get a lot of guidance in writing my Lister application. I wouldn't say I did it in a rush, but I didn't do it over. I completely endorse, approach your research grants, particularly in the current climate, exactly as was described by Chris, because it applies to all grants. And I had done, I suppose, a lot of the scientific groundwork for my Lister application before I started writing it. But I could see that it was about writing for a broad audience. I immediately saw it wasn't like a regular grant application, and I did that. But the most important thing in getting in the door is going to be your CV. And I get approached by a lot of potential Irish applicants, many of them excellent people, who it's just premature for them to apply to the Lister Institute. I would say that 90% of the people who've asked to go for coffee with me and have done, and we've gone through it, and I said, I do think you're potentially a candidate for this, but not for 2 or 3 years, right? Because one has to be practical about the competition, right? No matter how good your idea is, if you're not prima facie above a certain threshold, and we all know what that means, you're not doing yourself a service by spending your time on it. So, and I think part of what we need to be doing is more like what we heard from Birmingham, which makes me absolutely envious about how local support can create an easier stairway to being in a position to submit a credible Lister award. But at the moment, everything is just so difficult. And Cleona said something that earlier, and I, with huge affection for Cleona, when she said, for every success I've had 3 failures. Like, that's amazing. For every success I've had 10 failures. And increasingly we spend more and more of our time just trying to get funding, and this is part of the problem. It shouldn't be like this. So you want to go in with a 50% chance that you can be successful. So I think making your papers that you have as good as possible before you apply is crucial. It doesn't mean you need to have Nature and Science papers, right? It could mean that you have a series of Journal of Biological Chemistry papers that have established a unique niche for you, right? That equally works. But the second point, and this is where I think many excellent people fall down, people who are credible candidates, is that they have a great idea and they have a good CV, but their idea is not a fit for them, right? Even if you're stepping outside of your domain and you're doing something very creative, which Lister allows you to do, you jump with one foot, not both, right? One foot in what you know, one foot into a very new territory. That's about fit, making sure your CV fits the proposal.
I was just going to talk about the patient benefit and the public benefit, because I work a lot with children and their families and the questions that they want us to ask as researchers. It was a really fundamental part of how I developed my application. And some of the parts within the application were because our patient population were asking for us to do more work in uveitis in children with arthritis. But there isn't a lay summary part in the same way as there are in other applications. So I think you really have to be led by your research question about how you're improving patient and public involvement in my, yes. Julie?
I can comment on that, maybe from the scientific committee. We don't have, currently we do not have lay members on the panel. It's all scientists, and we typically don't seek that sort of impact. So it is judged on scientific merit. But for example, in Lizzy's application, which I remember, when you tell a story, a very clear story, interesting science, fantastic science, and then there's a patient benefit at the end, that really tugs at your heartstring. Then, you think, oh, good science, oh my goodness, and there are these poor kids who we might be able to do something to help. Then, double whammy.
I think so. I'm totally discovery, fundamental biology. Just like Julian, I work on DNA replication, okay? How the replication machinery is disassembled when it's done its job. You don't get, yes, in the future, maybe it has a chance, to make difference for the health, in next 20 years. But it is really totally fundamental biology. So, I cannot use that fact that, oh, poor children, you will help them, So I have to make it even more exciting and make it explain why it is really cool and why we really want to understand how it works, because we don't know. And it is really so important. And, you just have to make that extra step to try to make your research be of importance and be, of value to be funded.
Thanks. Just one thing that I have always found with when I'm reading and trying to help people apply for a fellowship, and this is specifically a fellowship, is that places like Wellcome and Lister are funding the fellow. So when you're writing that grant, do not pitch it like a UKRI or a Research Ireland grant. It's very much about you and developing you. And Lister will see that there's something special in you that they can then help to nurture and bring out. So writing a fellowship is written so differently to writing a research grant. And that's why you need fellows to help you and to read it for you. So that's just kind of one little tip that I think people forget.
And I would just add as well, coming back to clinical impact, patient involvement, you don't have very many words to lay out your science case. So I did have a little bit at the end of one of my first drafts, I think, where I kind of tried to tack on some sort of collaboration with pharma, and everyone who read it said, no, this is poorly developed. It seems tacked on. It seems like a passenger to then this excellent body of discovery research. So you should delete it. And I felt kind of a bit uncomfortable doing that, but it was in retrospect completely the right decision.
So having not sat on the other side of the table, I would only be able to answer half the question, or answer the question half as well as it needs to be. I think the problem extends not when the person starts trying to think about applying for Lister Fellowship. It probably starts 5 years before that. I have a rule in my lab that I will not hire a first-year postdoc if I don't think that they, in principle, can become a professor. I won't hire postdocs as technicians. Now, the problem is that in Ireland, and I think in the UK, there is still a laissez-faire culture about this. Now, that's okay if the person knows what they're getting in for. Maybe there's someone who's more cut out to be a staff scientist. But often they're actually extremely bright people who are not a good fit for the PI they're doing a postdoc with. And while they may be doing good work there, it's not enough career development for them, right? And so the problems that I see often it's not just about prematurity, it's about they have not been managing, or other people have not helped them manage their career trajectory sufficiently from the day they finish their PhD. You know, everyone can be forgiving about your PhD, but your postdoc has to be about developing yourself into a long-term sustainable career arc. That is the root cause, I think, of the problem. And I think some institutions are better than others at mentoring the population so that people don't find themselves trapped in these situations. I think in Ireland, we're not yet very developed about doing that. But on the positive side, you know, you don't have to go away to America and come back. I mean, I don't think I can say certain things that are currently embargoed, but some people have been very successful having never left Ireland. And I think that really is, you know, vindication of how much the Irish research ecosystem has developed over the past 20 years, partly because of people like Leona and others, and because of how much funding was put into science to develop our infrastructure up until the financial crisis. We are now seeing so many success stories that never left this island. And I think that's wonderful. I'm very proud of that as a citizen. So they are there too. But it's about finding the right career trajectory for the person when they're in an early postdoc, and that their papers, that they have some ownership of, such that it can be used as a platform for their own research vision and the ability to communicate that vision. Now, you have to be able to identify young people in your university, I believe, who have all those three germs, you know, who have great papers coming, who have a vision and need to learn how to communicate it. But if you have somebody who joined a random lab to do a postdoc that was relatively, you know, productive, got some good papers, but weren't really into that research field to the point where they have their own vision and the full confidence of their PI and their PI's network, you're already lost. And unfortunately, I see that for a large number of the postdoctoral fellows in Trinity, and it's not because they're weak candidates, it's because they haven't tailored their career structure.
If I can add something, I will be more that I agree, but I think there are kind of more from practical point of view. Yes, you can do things to improve your CV. It's not only papers, because when you're going for career development fellowship or Lister fellowship or something like this, it's not only the science that you delivered before. You have to show your proactivity. You have to show the ability, you know, that you will be able to lead a team. You know, there's a number of other things. So visibility, recognition in the field. So things that we advise people when we look, you know, so yes, well, of course we can say, oh, you need another 2 excellent papers, but that is really difficult to do, yeah? So, you know, the other things that, you know, you can do is like apply for small pots of money, whereas in your name, because that shows your proactivity of getting and being able to get independent money. Then other thing, go for conferences, give talks, so you can show that you are proactive and you are going and you are, you know, showing yourself to the world and you are visible in the world. You know, other things, you know, do some supervision of students because of in the lab, so you can say that you successfully supervised a student and that student later on got a PhD position, which is the development of others, which is also important part. You know, so there is now, especially with the narrative CV and, you know, the way that we want, there are all of these things that you need to have something to write about, and that shows a little bit more of a person when it comes to the panel, because when kind of sitting, let's say, on a welcome panel, when you look at somebody, and yes, they have excellent papers, but you know what, they've never supervised anybody, and now they want to have a team of 3 starting on day 1, and it's like, how are they going to do it? Yeah, and you know, so you need that kind of additional evidence of those different things that are important later on as a group leader, as a leader of the team.
Just to follow up, one other thing that I think for early career researchers, postdocs in particular, or early group leaders ambitious, that axis of people, is go to as many conferences as possible, but not just to say you've gone to conferences, to really become recognized in having mastered a craft. right? You are an expert in this one little niche, this one area that is an interesting question, and you can do this technique well. And about 5 people internationally who are respected know you can do this technique well. Because a big part of the lister is you actually get to suggest some of the scientific peer reviewers in the application. And so if you can say, I've got an international network of people who will probably all think, okay, this guy is quite good, and it would be good for the field if he continued working here in future, for example. That's kind of the reputation you want to be building in those early years, I would say.
Just to add one last thing to that, is just that it helps if people have heard of you. So just exactly what Chris has said. When I went for my lister, there was, I think, one member on the interview panel who had heard of me. Not that I knew him at all, but it helps if people know your name, which is what Chris is.
And we had feedback with some of our people that we mentor. They're going for coming back from interview, and the feedback was like, yeah, you have great CV, but why do we've never heard about you? So, very much what suggestion was from that, well, okay, so now apply for funding to go to a conference and get some, poster awards, because then you will write in your CV that you've applied for funding, tick, and you went for the conference, tick, and maybe you're actually going to chat to some people at that conference and make that network that, that is important. Yeah.
I think we need both. I think we need, hedgehogs and butterflies or grasshoppers. Like some people, I love interdisciplinary hopping, and some people of my best friends and some of the best scientists I know are just narrow structural biologists. There's nothing wrong with either approach. But when you go interdisciplinary, there's a tax, and there's the risk in getting the money, and there's the risk in doing the science. They're two very different things. Lister requires you to push the envelope a little bit, I think, in your vision, and mine was interdisciplinary. And they did the diligence on me when I was in my interviews. Why was I going into this field? What was the real motivation? How would I do it? It's a combination of, first of all, identifying whether where you're going is crowded or not. If it's crowded, you need friends to welcome you, or you won't be welcomed. If it's not crowded, which is often the case, then you just need to make sure it's feasible and that you won't fall over when you step over that gap. So there are different ways of persuading people about that. And the wonderful thing is, once you start doing it, I think it multiplies. So Lister may have started much of my development work, but all of a sudden, all my other funders were also contributing to it. They just didn't know they were. Because, that's how it happens. You need the initial confidence and indeed requirement to go into disciplinary, but then you find resources in other places. With respect to the science, just briefly, I think the way I explain this to people who are a little bit more junior than me, I say look at it from a perspective of regret minimization, right? The issue with risk in science, like in other fields, is we're always trying to minimize it or prevent it. But we shouldn't be wanting to prevent risk. You cannot be successful without it. What you should be worried about is, what if you risk something and could have been successful, but you never did it in the first place, right? So I think when you're doing something risky, you can, like putting money on a horse, you make the gamble in it, you make the decision in advance that could be lost. I could spend 2 years working on this, I'll survive. I accept that. But that is less traumatic to me than if somebody else does it and is successful at it while I did something of more moderate risk. So when I'm talking to my students as they're choosing their postdocs, I ask them to think about what is the maximum acceptable risk that they can stomach, and then to turn that into where they can leverage something that they're interested in, about not having regrets rather than having to avoid risk.
Lizzy, can I pick up on something you said earlier about how you pivoted your research to try and deal with a real patient problem? So how important were collaborations to you in putting your application forward?
As a basic scientist, everything that is possible in my lab is through really strong connections with clinical researchers. It was very funny because I actually wrote to fellowship applications that were unsuccessful because I tried to pivot into a different disease. So kind of showing, that I could move, but I didn't have the clinical research collaborative network that made it seem possible. And that was the large feedback that I got. My work with patients and their families is through the clinical research networks. I think if you are interested in clinically relevant questions as a basic scientist, you need to make sure that you have that network. It is also to do with, I've been at UCL for a long time, and it's taken me a long time to build up those collaborations through different hospitals. So there's also this question about moving and whether you should not move, but sometimes it's really fundamental. So it's just interesting about being high risk versus not high risk, but making sure, I think you have to have precedents that you're going to be able to succeed in that. And for me, showing that I have those clinical collaborations is incredibly important to set that precedent.
Any other questions? So can I just pick up with Aga and something you said earlier? You set up the Fellowship Academy. Something we were talking about before you spoke was about how much time you dedicate to that and how much your time you're, rewarded for dedicating to that.
Yeah, so kind of... So the Fellowship Academy, when I was applying for the job, I think for the role of being director of the Fellowship Academy, the job description said up to 2 days a week, and I thought, oh, it's not going to take so much. It does, okay? And I'd never had 2 days a week of free time before, so I'm definitely much more thinly stretched right now with my time, and I would like to have a little bit more thinking time about my own research, because that's what is really suffering, just kind of because I like talking, I like interacting with people, so I enjoy helping young people, and I like this job, but I think I'm probably looking forward to kind of taking it down a little bit so I can have a little bit time to think about science, because I actually really like that too. So it takes time, but I think I'm still hoping that once we, it's only 1.5 years since we started, so we have quite a lot of things in place that is organized, so more and more of those things can just happen by themselves rather than requiring a lot of organizational time. Once we've set up some of the support systems, and hopefully they will keep running. And I know that I will find new things to do, so and new ways of supporting early career researchers, so probably will fill the time anyway.
Any other questions on the floor? So what we're going to do now is we're going to break for tea, but obviously, we've brought Lizzy and Chris here over from England, especially to be able to network with you guys during the tea break and also afterwards when we're having the drinks. So please make full use of the fellows that were here to ask any questions that you may think of as we leave the room. So it's breaking for tea now. Coming back, it's not just tea, it's tea and homemade biscuits, Cleaner's request. And then we're coming back for a little session with the scientific committee, in which you'll hear some more details about how to apply and what the process is very quickly from Anne, and then more chance for questions that you may have for the scientific committee. So thank you. We'll see you back here at 4:00.